beautiful morning. On the behalf of Organizing Committee, NIPER Pharmacon 2022, I cordially welcome you all for the third and last day of this international conference. Today we are going to have two technical sessions going to happen in this venue before the lunch. Now we are here to listen to the first technical session on the very important area, therapeutic approaches for the CNS diseases. This session is chaired by Dr. Sasibala Singh. She is an eminent scientist, Director, NIPER, Hyderabad, and co-chaired by Professor S.A.S. Sharma from the Department of Pharmacology and Toxicology, NIPER S.A.S. Nagar. May I now invite both chairpersons, Dr. Sasibala Singh, Professor Sharma, to come on the dais, please, and do the needful. Thank you.
Check.
So, good morning and namaskar to everyone present here. I'm delighted to be part of this conference and especially this symposium on the third day of the conference on an important topic, therapeutics approaches for CNS diseases. I think we have realized that health is wealth, at least post-COVID and during COVID. And mental health are gaining a lot of importance. They're occupying a lot of space in healthcare system now for the reasons well known to us. You know, I always look at the system as, it's a computer. The software is the brain and the hardware is your body parts. So we need to take care of the software. If software is not in order, hardware will not function. So this is really good that we are talking on this issue today. And we know India also have, statistics also tells that neurodegenerative diseases are increasing. Even now, the epilepsy occupies the most of the space. Stroke, as I was talking to Dr. Gulati, has been from a conference on stroke in Singapore. India, sir, about 37% of the cases in the neurology is from stroke and then followed by headache, epilepsy, and other diseases. So we are looking forward to listening to very eminent speakers. We have 4 speakers who have done a lot of work in their areas. And our first speaker is Professor Anil Gulati. He is in Pharmazz company in USA, and I think he holds a very important and distinction in his field. He is MD, PhD, and is chair and chief executive of Pharmazz in Illinois. He's a professor emeritus in the University of Illinois. He has done a lot of work. I will not sort of come between him and my introducing him. I think we will listen to him, and his biodata is available in the Google. You can always browse and see. He's an outstanding researcher and an outstanding clinician also. Thank you, sir. Please.
Thank you very much for the kind introduction. It's always a pleasure to be back in NIPER Mohali. In India, this is one of the places where whenever I'm in the country, I do visit NIPER Mohali. So it's like my home. So what I'll do is, because I know that in the audience there will be few clinicians and many basics scientists, so I will go through a little bit slowly so that the clinical aspects of the the results which we got, they are also clarified and understandable. So I was professor and dean at Midwestern University, and based on my inventions, this company was started in 2010, and gradually it progressed and became bigger. Then I resigned from my deanship and professorship, became emeritus, but I still have an office at Midwestern, and still I'm a professor at UIC. But my salary comes from the company now. Next slide, please. I have the control. So, yeah. So I'm presenting results on behalf of many investigators. There were 18 centers in India who conducted the trial. And conflict is that I am the inventor and I am employee of the company which funded these trials. Okay. So slides are moving from there, or I have the control here? Yeah. Okay. Thank you. So this is the structure of the compound, which is initially it was called as IRL 1620. So this was made in 1988, late 88, 89 by Siba Gaigi from Japan. So when endothelin was discovered, first publication came out in Nature in 88, and the industry started working on endothelin A antagonist because it was described in that paper by Yanagisawa that it is the most potent vasoconstrictor substance known till date. So obviously the interest was, let us synthesize antagonist and see if we can treat hypertension. So that was a simple approach. So industry jumped on it. Thousands and hundreds of thousands of compounds were synthesized. And then ultimately in arterial hypertension it failed, but in pulmonary hypertension it succeeded. So today there are several drugs based on endothelin A antagonist in treatment of pulmonary hypertension. But endothelin B receptors were completely ignored. And we started working in 89 on endothelin B receptors, and we recognized, and others recognized, that there are very, very high concentration of endothelin B receptors in the brain. So our strategy was also to see whether endothelin B receptors in the brain are controlling blood pressure. And we conducted a lot of studies and found negative results. We gave IRL 1620, which is a highly selective endothelin B agonist in the brain. Nothing happened. So peripheral, there was some dilatation, and which was transient, and then after that, so not much of cardiovascular effects were observed. So then after that, many years of research of failure, one idea struck that these endothelin B receptors are present on the endothelial cells and they are dilators. So we thought, when the tumor is growing, why not give this substance, and when the fastest part of the growing tumor will be lacking smooth muscles which are constrictor, but will be having more of endothelial cells which are dilator here by ETB. So we gave, and we found that solid tumors, there was increase in blood flow. So we discovered and patented the technology that we can use 1620 to dilate the tumor and give chemotherapy 15 minutes later and enhance the delivery of anti-cancer drugs to the tumor. So that technology was of interest when I presented at one of the cancer meeting, and Spectrum Pharmaceuticals, they came, they did their due diligence and licensed the product. And so they named it as SPIS 1620. So they did phase 1 clinical trial, 32 patients in cancer patients, and then they did 2 phase 2 trials. One was in non-small cell lung carcinoma, and the second was in cholangiocarcinoma. Both were very tough cancers, so the trial failed. So it was back to the bench again. So then we kept on our research on endothelin B with focus that there is very high concentration in the brain, So roughly about 2002-3, there were some publications which started coming out that there are hidden stem cells in the brain, and there are these hidden stem cells can, when the brain gets damaged, they tend to form new neurons. So that was the idea which came, and ultimately we and there is a scientist Koyama from Japan, they found that endothelin B receptors, if they are knocked out in the brain does not develop and the animal dies. So the idea came to us that, and in collaboration with Advocate Lutheran Children's Hospital, neonatology, I gave them the project, their fellows, the project that let us see the characterization of endothelin B receptors during growth. So in a rat, newborn rat, till 1 month of age, we kept on every week. And we found that when the rat is born, number of neurons are very high, and then by 28 days, it goes down to almost half. And it correlated perfectly well with endothelin B receptor concentration. So our interpretation was that endothelin B receptors are integral part of development of central nervous system. So then we thought that if they are that important, why not give endothelin B agonist and see what happens to the brain. So in a normal animal, nothing happened. But in a damaged brain, we found that endothelin B receptor expression increased. So we thought, let us target stroke or cerebral ischemic stroke. And 2008, I was in Nipur that time, and Dr. Shamsundar Sharma was having the model of middle cerebral artery occlusion. So we tried some experiments, we got some, but I was about to leave, so we continued those experiments in US and ultimately found that there was very significant reduction in infarct volume. You can see that middle cerebral, almost 90-95% reduction in infarct volume. And tunnel cells also. So we did a lot of studies. They have been published in Brain Research, scientific reports, and many other journals. There are almost like 15-16 papers, basic indicating that endothelin B receptor stimulation helps in the animal model of stroke. So then we thought that let us find the mechanism, how it works. So one mechanism which was very conclusive is anti-apoptotic phenomena. So prevention of cell death was taking place, and the second aspect which was of interest was multiplication of neurons or formation of new neurons. So these are some of the results which are indicating that double cortin, neuro-D, HUC, HUD with sovateltide. Now let me introduce the name sovateltide. Since the drug IRL-620, there was no proper name. So once it came to a stage that the drug has to go to clinic, you have to get a proper name. Now the procedure to get a proper name is that you have to get approval from WHO. There is a committee in WHO which gives the classification of the name, and that committee meets 2 times in a year. And then there is a committee in US, USAN. So we applied to WHO and got the name sovateltide approved, and then we applied to United States AN, nomenclature committee, and the name was approved as sovateltide. So now official word worldwide is sovateltide. That is that. So now you can see these figures that there is highly significant increase in neuro-D, HUC, indicating that these are the markers for neural progenitor cells which are improving. So stem cells, endothelin B agonist sovateltide is not acting on stem cells in the brain, but is acting on one step after the stem cells, that is neural progenitor cells. Now, these findings are more on mitochondrial fission and fusion, and once again, you will find that apoptotic phenomena, it is acting through prevention of cell death and mitochondrial preservation. So all findings are indicating that mitochondrial preservation is quite significant at day 1 and day 7 of post middle cerebral artery occlusion with sorbitol. So these are some pictures indicating mitochondrial DNA activity. So you can see the green bar, significant improvement in mitochondrial DNA activity in presence of sorbitol. Then these are some electron microscopic pictures, once again indicating that mitochondrial preservation is quite intact, and mitochondria is the seat of activity that is very important in the brain. So cross-sectional area, so all these findings are indicating that the mechanism of action is twofold. One is that it is protection, and the second is neuroregeneration. So protection, we published results that it is anti-inflammatory, antioxidative, apoptosis is decreased, mitochondrial biogenesis, so it will preserve neurons and functional recovery. The second part, which is a little bit delayed, is the angiogenesis, which correlates with VEGF factor, DXN, NeuroN, and the mechanism is through neural progenitor cells. And we also found that presynaptic, postsynaptic markers are also increased. So these findings also have been published in scientific reports. So these are some of the publications, key publications which you can look at. So in nutshell, what is happening is that besides prevention of apoptosis or anti-apoptotic activity, there is stimulation of neural progenitor cells and formation of mature neurons. So we quantitated, it is close to 300% increase in formation of mature neurons by sorbitol. So we did phase 1 study. We got the approval from DCGI here to conduct phase 1, and phase 1 came out to be okay, although we had done 32 patients plus close to 200 patients in cancer, but the committee decided that you have not done in normal individuals, so do it again. So we did phase 1, came out okay, and then we started on phase 2. We got the permission to do phase 2. These results have been published in CNS drugs, and that came out to be quite promising in the sense that the neurological outcome was improved. And we all know that stroke has faced so many failures that almost every drug which has been developed since TPA has failed. So nothing has come out. So we were well aware of all that fact, but the results were so promising, we decided that let us move forward and spend the money to get phase 3 permission. So after phase 2 results were published, we embarked on phase 3. We got the permission to do phase 3 trial, which was done in 18 hospitals across the country. And this was the design of the trial. So the design of the trial is such that NIH S.A.S. is the assessment of the extent of stroke. So we took moderate stroke, that is above 5, so 6 and above NIH S.A.S. was kept those patients, and we excluded patients wherever any surgical intervention was there. For example, thrombectomy and those kind of things, they were excluded. So intracranial hemorrhage was also excluded. So only ischemic patients. And the reason is, although we know that the drug is neuroregenerative and all that, so it can act, but to do a trial, you have to be, better it is to keep it clean and not too many interferences. So that was the main reason why we wanted to have only ischemic stroke. And so those patients were included, randomized standard treatment, whether they were blood thinners or TPA, they were allowed to be given. So 2 groups, saline and PMZ 1620 or sorbitol, and visit 1, 2, 3, 4, 5. So 90-day assessment, that is the gold standard which all the regulatory agencies across the world look for. And so primary endpoint they look for is 90-day, and nowadays US FDA and Japanese agencies, they are looking for longer term also. But primary endpoint still is 90 days. So we did that trial. There were 760 patients who were assessed, and we did not want any recurrent stroke. So All India Institute Delhi, they got a lot of patients with recurrent stroke, they were excluded. So only fresh stroke patients were included, and they 158 patients, 78 in control, 80 in sovatiltide, and 90-day follow-up completion was done by 70 patients in control and 67 in sovatiltide. So the mean age, body weight, male-female ratio, they are very similar. Now very important is NIH, S.A.S at baseline, 9 and 10 median, they are very similar to each other. Median aspects is by radiologically, you can find out the volume of infarct by three-dimensional CTs and MRIs. So you can see 8 and 8, exactly similar volume of infarct was there. Thrombolytic therapy, which is TPA, which is the only drug which is approved, has to be given within 4.5 hours, because the later you give, the chances of intracranial bleed is very high. So recommended is 4.5-hour limit. So you will see that 25% of the patient in control got thrombolytics and only 11% in sovatiltide got thrombolytics. And that is statistically different. But I want to highlight that in spite of the fact that more patients got in control thrombolytics, but you will see the results are in favor of sovatiltide. Similarly, large artery atherosclerosis is the most devastating, and high mortality is there with large artery atherosclerosis and more disability. So more 46% patients in sovatiltide compared to 37, although statistically not different, but still 10% difference. The patient's outcome in sovatiltide was better. Median hour interval is about 18-19 hours after the onset of stroke. So that also is very promising in the sense that instead of 4.5-hour window, we kept a 24-hour window based on our animal experimentation data. So in the trial, it is roughly about 18-19 hours. So the subtypes of stroke, that is also important, because we do not want that the tilt is more towards devastating in one, but here it is opposite, that large artery atherosclerosis, there are more patients in sovatiltide than in control. But other small vessel occlusion is more or less is the same. Now comes the result. So the results are neurological assessments, and the gold standard which US FDA and other regulatory agencies look for is head-to-head comparison of modified ranking scale. So these are neurologists, they rank according to the neurological tests, and modified ranking scale at day 90, head-to-head comparison, p-value is 0.007. So then we thought that let us analyze from baseline how many patients are having more improvement in modified ranking scale. So modified ranking scale of 2 or more improvement from baseline to 90-day, once again is statistically significant, 0.0045. Another thing, with our initial correspondence with US FDA, they said that baseline modified ranking scales are not reliable. So can you assess patients with 0 to 2 modified ranking scale? 0 to 2 means that 0 is normal, 1 is clinically, the disability is not even obvious clinically, and 2 is very mild clinical disability, and then 3 is that you need assistance in walking and all that. So 0 to 2 is something which is very important. So you can see that patients at 90-day with 0 to 2 modified ranking scale are 54 compared to 39, and p-value is reaching at 0.001. Then another way to test is NIH S.A.S. So although this is not a primary endpoint with US FDA, a modified ranking scale is the primary endpoint, but NIH S.A.S. also at 90-day head-to-head comparison is coming out to be significant, p-value 0.002. Then number of patients having 6 or more improvement in NIH S.A.S. is also significant, 0.1. Barthel index is another way to assess, and in Barthel index there is a component of cognitive also within, so the head-to-head comparison is 0.1, statistically significant. Now change in Barthel index of 40 or more missed the level of significance, 0.6, but still there is a from 61% to 76% number of patients in control and sorbitol-tide. Then we thought that let us see how many patients are having full recovery. So full recovery with all 3 parameters, that is modified ranking scale, NIH S.A.S, Barthel index. So modified ranking scale 0 is full recovery, NIH S.A.S 0 is full recovery, Barthel index 100 is full recovery. And at the bottom you can see the percentages from 15 to 26, 25 to 37, and so on. So at least 10% more patients are having full recovery in the sorbitol-tide group in any way you measure. Then quality of life index, although these are exploratory endpoints, they are also coming out to be statistically significant. Montreal cognitive score did not come out to be significant, and the reason for this is for cognitive, in Alzheimer's disease we are doing a trial, but there the drug has to be given every month. It's a longer term, so the dosing has to be different. So in stroke, the dosing was 6 days. On day 1, patient got 3 doses of sorbitol-tide. On day 3, patient got 3 doses, and on day 6, 3 doses, and that is the end of treatment. But in cognitive, we have to give every month. Survival mortality log ranking Cox shows no statistical significance, and there were 4 deaths in sorbitol-tide and 2 in control. And once again, if you notice large artery atherosclerosis, 3 patients who died were having large artery atherosclerosis in the sorbitol-tide group. These are the adverse events which have been recorded. So 33 adverse events in 24 patients and 27 adverse events in 15 patients, and most of the adverse events have not been correlated with the drug. So basically safety was established in phase 1, phase 2, and now in phase 3 also. Now it is a lot of interest that how many patients got intracranial hemorrhage, because that is one of the major adverse events which happens with TPA. So 7 patients out of 80 in sorbitol-tide group and 7 patients in 70 in the control group got intracranial hemorrhage, which is similar, and percentage-wise also about 8.7, 8.9% is equal to the conventional reports which have been published across the world. So there is no incidence of intracranial hemorrhage, and we also did in vitro clot lysis and all those studies, absolutely no interaction with TPA, and it can be safely given with TPA. So the summary, you can see that any way, with any parameter we analyze, this compound sorbitol-tide is showing highly significant improvement in neurological outcome. So p-value of, you can see that on the other side. Neurologists are always concerned with what is the shift which is taking place. That is clinically meaningful improvement in neurological outcome. And you can see the shift of modified ranking scale from 0 to 6. 6 is death, and 5 is severe disability. So the shift, that means from control 15 to 25% shift is taking place with patients which are completely normal, that is no disability. And with modified ranking scale 2, from 54 to 76%,